Hi there. This is going to be a short video on the topic DNA. Topic DNA falls within the larger topic biochemistry or molecules for AS module AS1 module 1 biology. DNA is a very important topic, not only for questions in this particular module, but also with respect to upper sixth work. There's a whole section of Module 5 in upper sixth which deals with DNA and protein synthesis and DNA technology and genetic engineering, as it's sometimes referred to as. So it's quite important that you get a good grounding, good understanding of the basic structure of DNA and other features relating to DNA at this level before you proceed to upper sixth. So to get the most benefit from this video, you probably need access to a set of my notes on the DNA topic and a set of past paper questions. I'll be making constant reference to the notes and to various pages and to particular questions as we go through this topic. So it'd be useful if you did have a set of notes and a set of questions in front of you. If you need a set of notes and set of questions, then you'll see my number there. You can get in touch, and I'll furnish you with those. Now, where do we start with DNA? I think it may be useful just to start with something that I looked at in another video. By the way, there are other videos which deal with other aspects of biochemistry. There's a video dealing with carbohydrates and lipids, and there's one I've done recently on proteins, so you can access those as well. So the starting point maybe for DNA is to recognize that DNA is part of a larger group of molecules called nucleic acids. And essentially, as far as you're concerned, the two major nucleic acid molecules are DNA and RNA. And there are several different forms of RNA, and that's something we're going to look at in a moment or two. Something that's useful just initially is to talk about what elements are present in the DNA, in the DNA molecule. And for this, I would maybe have a look at this table. And this table is going to compare carbohydrates, lipids, proteins, and DNA. So we look at carbs here, lipids, proteins, and I'm going to refer to DNA as nucleic acids, N-U-C-L-E-I-C, nucleic acids, so that includes DNA and RNA. And what I just want to look at initially is what elements are present in these molecules. And you might already know that carbohydrates, lipids, proteins, and nucleic acids, they all contain carbon, hydrogen, and oxygen. So carbon, hydrogen, and oxygen. So each of them contains carbon, hydrogen, and oxygen in their structure. And then proteins also contain nitrogen. Okay, so proteins also contain nitrogen, and as do nucleic acids, and nucleic acids also contain nitrogen. DNA also contains nitrogen. And there's another molecule, then another element that's considered, that's phosphorus. And here we find that nucleic acids also contain phosphorus or phosphate. So nucleic acids DNA contains elements carbon, hydrogen, oxygen, nitrogen, and phosphorus or phosphate. And that's a useful table to be able to reproduce. Sometimes you get questions in the biochemistry talk that ask you to be able to distinguish between different molecules according to what elements are present. So DNA then contains the elements carbon, hydrogen, oxygen, nitrogen, and phosphate. And now I'm just going to look then at the structure of DNA, at the more detailed structure of DNA. Well, from GCSE, you learned that DNA was a double-stranded molecule, like had a ladder shape, like the shape of a ladder, and essentially you have the two. So these are the sort of the sides of the ladder, as it were, and you recognised that the most important aspect of the DNA molecule was actually the bases. Then you had bases A matched with T, and C matched with G, and G matched with C, and so on, and G. C matched with G. Okay, and that was something you learned. You learned at GCSE. C and G matched with C, and C matched with G, and so on and so forth. So the most important aspect of the DNA molecule was actually these bases. It wasn't the outsides of the ladder that were important. It was as it were, the rungs of the ladder, which made up of the bases. And I think you've come to recognize that it's the sequence of the bases that is important. It's the sequence of the bases, the order of the sequence of the bases. And you probably already made reference to that with respect to the structure of proteins. Because when you're looking at the structure of proteins and looking at how a cell made its proteins, you recognize that DNA was involved in the cell making proteins. And how DNA was involved was that DNA provided the code, or the sequence of bases provided a set of instructions or a code to the cell, telling the cell the order in which it should join together its amino acids to make its polypeptides. So the sequence of amino acids in a polypeptide determines the final protein that's going to be produced. And the DNA codes for that sequence of amino acids in the polypeptide. So in a way you can say that the DNA code is a code for the primary structure of a polypeptide. So the DNA molecule codes for the primary structure of a polypeptide, and in that way DNA effectively codes for the proteins, So the DNA code tells the cell, as it were, what proteins to make, the different proteins that it will make. So if you're looking then at my notes, I'm keeping my eye on the notes so I don't miss anything here as we go along. You'll see that I said DNA, about a third of the way down the page, I've said bullet point 3, I've said DNA acts as a code as it contains information needed by cells to make the cell's proteins. And I've also said and enzymes. And of course enzymes are proteins. So the cell can only make its enzymes correctly if it's got the code, got the DNA code. And of course enzymes are absolutely vital to a cell then, because the enzymes dictate or control the chemical reactions that happen in the cytoplasm of the cell. So the DNA is essential in telling the cell the correct enzymes so that it can have the correct chemical reactions taking place inside its cytoplasm. Okay, so another term then relating to DNA you might come across is the idea of what is a gene? What is a gene? And you would associate genes with DNA. Well, in simple terms, if, for example, we consider this here is the DNA molecule. Now, this is only single-stranded, but if you can imagine that it's a double-stranded DNA molecule, the DNA is often stored on things called chromosomes, stored on chromosomes. So I'm just making up what we might imagine to be a chromosome. And a chromosome essentially is a device in which, or a device, a mechanism or a system in which to store DNA. So there's the DNA stored. This is a very simplistic version of what a chromosome is. DNA stored on a chromosome. Now, then the term gene. Well, a gene simply refers to a short length of DNA. So I'm just removing a short length of the DNA. So one gene, that might be one gene there, that's present among in the DNA. So effectively, the DNA on a chromosome is just a whole series of genes. So that's one gene, and the next gene will be there, and the next gene, and so on and so forth. So the DNA molecule array on a chromosome consists just of a whole series of genes. One after, and a definition of a gene then is a short length of DNA with sufficient code to code for a single polypeptide. That's the definition of a gene. So a gene is just a length of DNA, a short length of DNA. The bottom page one, DNA is a length of, a gene is a length of DNA sufficient to code for the making of a single polypeptide. Okay, now, and then over the page I'm saying a particular protein may consist of several polypeptides in its structure. And you come across this idea that proteins may have quaternary structure, and that simply means there's several polypeptides present. And if there are several polypeptides present in the structure of a protein, then you may need several genes to code for that protein, because you may need a separate gene for each of the polypeptides if the polypeptides are different from each other. Okay, so the DNA molecule then, a long molecule with two strands, as it were. These are sometimes referred to as the backbone of the molecule, these two strands, and these here would be the bases in the center. So the DNA is a long thread-like molecule. Now, these bases, if we look at the bases more closely, we see that, or look at the structure of DNA, we find that DNA is made up of, DNA is a big molecule, sometimes referred to as a macromolecule or a giant molecule, and it's made then up of smaller molecules or subunits, and the subunits in the case of DNA are referred to as nucleotides. Nucleotides are the small molecules, or the smaller units that make up the DNA molecule. And if you have access to my notes, then you can see illustrations of that. But basically, the subunit for DNA is a thing called a nucleotide. And a nucleotide itself is made up of three components. It's made up of a phosphate group, which is their phosphate group. And this molecule here is a 5-carbon sugar called deoxyribose. So we're talking DNA here. That's the D in DNA is the deoxyribose sugar phosphate group. And then this in the center could be a base. So this could be, for example, the base A, which stands for adenine. Okay. So there's a nucleotide. And as you might appreciate, that there are 4 different types of nucleotides. 4 different types of nucleotides. And they're only different in terms of the base. So they all have this shape, and they all have a base here, but the base is different. So you have nucleotides, DNA nucleotides that have an adenine, an A there, or a T, which is thymine, or a C, which is cytosine, or a G, which is guanine. And unlike at GCSE, where you just were happy enough with just the letters, at the AS level you need to know the names of the bases. So you need to be able to spell them correctly as well. You may ask you to write down the name of a base. If you spell it wrongly, then you mightn't get the mark. So those are the 3 components then that make up a nucleotide, and it's a whole chain of those nucleotides that make up one side of the DNA molecule then. And if you look closely, then you'll see that where the different, I said at the start that you have carbon, hydrogen, oxygen, nitrogen, and phosphorus. All these elements are present in DNA. Well, the carbon, the hydrogen, and the oxygen are present in this molecule here, which is the deoxyribose, D-E-O-X-Y-R-I-B-O-S, deoxyribose sugar that contains carbon, hydrogen, and oxygen. The nitrogen is present in the base. The bases are often referred to as nitrogenous bases. There's a lot of nitrogens here, and there's the phosphorus in the phosphate group. So there's the elements that are present in DNA: CHO, carbon, hydrogen, oxygen, nitrogen, and phosphorus. All those elements are present in in a nucleotide. So DNA is then made up of subunits called nucleotides, and you'll see there in the notes the 5 different types of nucleotides. Now, to make the DNA molecule, each nucleotide is joined to the one beside it. So you sort of see it in that diagram there. You can see a series of nucleotides. The thing circled at the bottom there that's one nucleotide, and then you can see other nucleotides stacked above that. Nucleotides themselves are joined together by condensation reactions. In other words, the uprights of the ladder are joined together by means of by condensation reactions. And that's reactions between individual nucleotides. So nucleotides are joined by condensation reactions. And the bond created there is referred to as a phosphodiester bond. A phosphodiester bond. Okay, phosphodiester bond. So nucleotides joined together by phosphodiester bonds in condensation reactions. Okay, now where do we go from there? We also maybe refer to the term complementary. Complementary would refer to the fact that in the DNA molecule, in the 2 strands of the DNA molecule, you would always have an A base and a T base matching with each other. So if this is the other strand of the DNA, adenine and thymine are said to be complementary bases. That simply means that they have the same shape as well. They have the same shape, but they have shapes that fit together. So they fit into each other. Adenine fits into thymine, and cytosine fits into guanine. They're said to be complementary bases, and they're held together. The bases are held together by hydrogen bonds. So you get hydrogen bonds effectively holding the 2 sides of the ladder together because the hydrogen bonds are between the bases. And if you wanted to separate the 2 strands of the DNA molecule, then by those hydrogen bonds that would break would allow the 2 strands to move apart, to be separated apart. Okay, the whole molecule then, the whole DNA molecule, once you've got the DNA molecule, it's then twisted into what's called a double helix shape. That's more a GCSE question. They'll often ask you what shape is the double-stranded DNA molecule, and you see it's got a double helix shape. So that's DNA. What about the other nucleic acids that you need to know about? Well, the other nucleic acids you need to know about are RNA, which stands for ribonucleic acid, RNA. And RNA has 3 different forms. You have 3 different types of RNA. Essentially, RNA is a single-stranded molecule. So there's an RNA there. RNA is a single-stranded molecule, and it will have a sequence of bases as you move along the RNA molecule. It also has a sequence of bases. So maybe G there, and then C there. But notably, the RNA molecule doesn't have a thymine base. Instead of having a thymine base, it would have a uracil base. So the difference between the DNA and the RNA, RNA is just a single-stranded molecule. And RNA has got one base which is different. RNA's got a cytosine, a guanine, and adenine, but it doesn't have a thymine base. It has a uracil base instead. So there's 2 major differences between DNA and RNA. DNA is double-stranded, RNA is single-stranded. DNA has bases adenine, thymine, cytosine, and guanine. RNA has bases adenine, guanine, cytosine, and uracil, U-R-A-C-I-L. RNA is a single strand. The other thing with RNA then is RNA, 3 different types of it, and the 3 types of it are referred to as messenger RNA, transfer RNA, and ribosomal RNA. So I'll just write those 3 down. So the 3 types of RNA are messenger RNA, and messenger RNA is written with a small m, like that. mRNA, messenger RNA, and then you have transfer RNA, which has got a small t, transfer RNA, tRNA, and then lastly rRNA. Now, these RNA molecules, in simple terms, are involved in the process of protein synthesis. When the cell is making its proteins, when the DNA code is being read, and the proteins are being made inside the cell, the RNA molecules are heavily involved. And I maybe just want to illustrate that just quickly for you. So you've got 3 types of RNA. Okay, I want to illustrate that just briefly here. So these different types of RNA are involved when the cell is making proteins. Now, the thing I should say to you about this is you don't need to know any detail about this much, because this is dealt with as a separate topic in Module 5 in Upper 6th. The topic is called protein synthesis and explains how DNA and RNA and everything are involved in making proteins. But just briefly, I'm just going to look here at a cell. Let's see if you can. It's a cell. So let's say that's your cell. And this is the nucleus. I'm going to do the nucleus like this. Okay, this is the nucleus. And these gaps in the nucleus membrane are nuclear pores. Okay, and inside the nucleus then you would have DNA. So I'm just going to illustrate DNA inside the nucleus like this. So just illustrate DNA like that. So there's the double-stranded DNA molecule. There we are. And now, during protein synthesis, proteins are made in the cytoplasm, and the proteins are made out in the cytoplasm at something called the RER. And the proteins are specifically made of things called ribosomes. So there's a bit of RER, and these little fellows on the surface are the ribosomes. So the code, or the code for the proteins that the cell is making. So this is a cell. This is the big picture of what's happening during protein synthesis. I'm really just showing you this just to sort of give you some idea about the roles of the different RNA molecules when the cell is making proteins. But as I say, you don't need to know the detail of this, because this is for upper 6th. But you would need to know just a wee bit just about the RNA molecules, what they do. So first of all, this is the DNA inside the nucleus here. DNA. And as I said before, a gene is a short length of DNA. So I'm just going to pick that little piece of DNA there in the middle. So this is really just an illustration. So there's a gene there, a double-stranded piece of DNA. And let's say then the cell is making a protein out in this is the RER. The cell is making a protein out here at the RER for which this gene codes. Well, the code's in here. The code for the genes, the code for the proteins in here, but the protein's made out here. You can't take the DNA out of the nucleus. There's a couple of reasons. The DNA molecule is much too big, and it's also attached to something called histone proteins. And we'll look at that again. So the DNA can't leave the nucleus, but somehow or other you need to get the message or the code out to the RER where the proteins are made. And what happens essentially is that the DNA molecule is a copy is made of what's called the coding strand of the DNA molecule. One of the 2 strands of the DNA molecule is actually contains the base sequence or the code. And that copy of the DNA molecule is referred to as messenger RNA. So you get this molecule called messenger RNA. So there it's there, mRNA is made. And then the messenger RNA, what the messenger RNA does is it leaves the nucleus. It goes out through a nuclear pore, the messenger RNA, and it goes to the ribosome. It goes to a ribosome. So I'm just going to bring it over here and attach it to that ribosome. So there's the messenger RNA attached to that ribosome. So basically the messenger RNA is a kind of a copy of the original gene, copy of the DNA that's inside the cytoplasm. Messenger RNA is small enough to get out of the nucleus through a nuclear pore, and the messenger RNA goes and attaches to a ribosome. So that's the role of the messenger RNA. What about rRNA or ribosomal RNA? Well, these ribosomes are made of ribosomal RNA. So that's the role of the ribosomal RNA. They're basically, they make up the structure of the ribosomes. So that's ribosomal RNA. And then there's another molecule called a transfer RNA. And a transfer RNA is essentially a molecule which is involved in bringing amino acids into position. So this is the transfer RNA molecule here. I'm not doing any detail. The transfer RNA molecule, tRNA, is a molecule that's involved and bringing amino acids into position during the making of a polypeptide. And that's sufficient at this stage to know about those. So the messenger RNA is a copy of the gene. It leaves the nucleus and goes out to the RER, out to the ribosome. The ribosome is made of ribosomal RNA, and the transfer RNAs or tRNAs are involved in transporting. I suppose you could say T for transport. They transport the amino acids into position during the making of the protein. So that's the role of those three different types of RNA. So, the other thing I should have said about RNA, the third thing about RNA is that instead of having deoxyribose, so the nucleotides for RNA, instead of having deoxyribose sugar, they've got a different sugar called ribose sugar. Okay, you can look in the notes there, you'll see a table that compares DNA and RNA. It basically says DNA is a very large molecule, it's double-stranded, it contains the base adenine, guanine, cytosine and thymine, and nucleotides contain the sugar deoxyribose, whereas RNA is a much smaller molecule, single-stranded, contains the base uracil instead of thymine. Uracil could contain the base uracil instead of thymine, and the nucleotides contain the sugar ribose instead of deoxyribose. Okay, and essentially that's the role then of DNA and RNA in protein synthesis. Now, the next thing I want to look at, which comes up quite a lot, is DNA replication. Now, DNA replication simply means that DNA copying itself. DNA can copy itself. Just maybe before we look at that, just a few questions. So question one, pretty straightforward there. You're identifying the different parts of the DNA molecule. Question two, you also see the structure of DNA there. You're describing the structures 1 to 4. And you have a few more such questions there in question 4. That's the structure of DNA. Question 5, the structure of DNA a bit. And question 6, a little bit about DNA as well, and so on. So the structure of DNA does get asked about, but not in such great detail. But the other thing it does get asked about in some detail in this topic, or it may also be asked in another topic called continuity of cells, which is essentially mitosis and meiosis, is how DNA replicates itself, how DNA makes a copy of itself. And that relates to cell division, mitosis and meiosis. Why does DNA need to make a copy of itself, and where does this happen? Well, the answer is it happens inside the nucleus. DNA is present inside the nucleus. And before a cell can divide, which effectively cell division refers to how a single original cell can go through a process whereby it can create 2 cells. The cell is said to divide into 2 cells. And that cell division can be either mitosis or meiosis, irrespective of whether it's mitosis or meiosis, which is dealt with in more detail in the continuity topic. One thing that has to happen in the cell before a cell can divide is that the cell has to copy all of its components. It has to copy all of its cytoplasm, all of its organelles, and of course it has to copy all of its DNA. If a cell is going to, as it were, make 2 cells, then it needs enough DNA for 2 cells, and that involves DNA replication. And so we want to look at how DNA replicates itself. Now, I'm not going to draw stuff on the board. It's just too complicated. But you see there on page 7, my notes, there's a good illustration of DNA replication. It's also illustrated quite well in question 2 in the question booklet there. It shows the DNA replicating. And essentially, they'll ask you questions about, they might ask you for 4 marks just to describe the process of DNA replication. And essentially what's involved is, you start off by saying that for the DNA molecule to replicate itself, the DNA molecule essentially being like a ladder shape with bases in the center. Essentially what has to happen is that the 2 strands of the DNA molecule have to come apart. The 2, as it were, uprights of the DNA molecule come apart, so you separate the two strands. And to do that, you need to break bonds between the bases. So let's say you have an A base there and a T base there. Between the A and the T base, you'll have what kind of a bond? You have a hydrogen bond. And the same with all the other bases in the DNA. There are hydrogen bonds between the complementary bases. So the first step in getting the two DNA strands apart is to, and often this process is called unzipping. It's carried out by an enzyme called DNA helicase. The DNA helicase causes the hydrogen bonds to break, causing the two strands of DNA to separate or unzip from each other. And that's the first stage. And then what happens is that alongside each of these two separated strands, you get new DNA molecules lining, new nucleotides sort of line themselves up alongside the two separated strands. You get nucleotides line themselves up there. So effectively you're creating two DNA, eventually then these nucleotides join together, and basically you've created two DNAs. Original DNA, now you've got two DNAs. So the lining up of these nucleotides is brought about by another enzyme called DNA polymerase. DNA polymerase, this is the second enzyme. And essentially what it involves is you have what are referred to as free nucleotides. These free nucleotides come into position alongside the two separated strands. And often you can refer to the two separated strands as two template strands. Each of the two separated strands acts as a template alongside which free nucleotides line up such that you have complementary base pairing. Once the free nucleotides are lined up, then free nucleotides they bond, or they attach by hydrogen bonds to the nucleotides of the DNA strand. But here's the key point. The DNA polymerase causes the free nucleotides to stick to each other, to join to each other by condensation reactions to form phosphodiester bonds. And that's important. The enzyme DNA polymerase causes the free nucleotides to attach to each other by phosphodiester bonds in condensation reactions. So the DNA polymerase encourages the making of phosphodiester bonds. Okay, very, very important point. This process, this way in which DNA copies itself or replicates itself is called semi-conservative replication. Semi-conservative replication, and you get many questions on this. So you often get asked, what do you mean it's called semi-conservative replication? And the two marks to describe that essentially is, what you say is, when DNA is replicating itself, each of the two, these are called parent strands. This is called the parent DNA, if you want to say. Each of the strands of the parent molecule act as templates to create, on which free nucleotides join up to create the newly formed strands. So DNA semi-conservative replication of DNA, where you end up with two new DNA molecules that are both identical to the original DNA molecule. Each new DNA molecule consists of a template strand, those are the template strands there, template strand, which comes from the original DNA molecule, plus a newly constructed strand made from free nucleotides. And that's the definition of semi-conservative replication. And you frequently get asked questions then about DNA replication. So if you're looking there, typical question 3 in the booklet, it just says, describe the process of DNA replication in cells. And essentially it's looking 4 marks. And you can go through the points there and get the 4 marks quite easily. As I say, this has overlaps with the cell continuity topic, and you might be asked this in relation to a meiosis question. Now, the last part of this topic that I want to look at, and probably the bit that gives students the most worry sometimes, is questions to do with the Mendelssohn-Stahl experiment, sometimes also referred to as the experiment with N15, N14 stuff in it. And students often get very worried about this kind of question, and they simply find it difficult just to understand it. Now, essentially, what's happened here is this was an experiment done by 2 guys called Mendelssohn and Stahl, And what they did was they set out to prove that this is the way that DNA replicates itself. This is how DNA does replicate itself. That DNA does replicate itself by this process of semi-conservative replication. Now, students often wonder, well, what do you need to prove this for? Because surely this is obvious. This is the way that DNA would replicate itself. But if I was to say to you that scientists cannot observe DNA replicating itself, they only can come up with a theory or an idea about how it happens. And essentially, here's where they're thinking. If you had an original DNA molecule like that, so that's the original DNA molecule. If the cell needs two copies of that molecule, well, here's what could happen. What could happen is what we've just described. The two strands separate from each other, and then you get the two. I'll just do the two new strands. You get the two new strands made alongside each other, and that's referred to as semi-conservative replication. So this is like the obvious way that DNA would replicate, and this is what these guys were trying to prove, because they were thinking, well, there's another way it could happen. Imagine there's the original DNA. If the cell wanted two copies of that DNA molecule, what it could do is just hang on to that copy, that DNA molecule like that, and then make a brand new DNA molecule that consisted of pre-nucleotides. Didn't draw that very well. In other words, and this is very different from semi-conservative replication. So this is semi-conservative replication. Semi-conservative, the idea being that in the new DNA is semi-conserved. That means half in the new DNA molecules, half of each of the old DNA molecules is kept or conserved. Whereas this kind of replication here, we don't have a name for this, but this looks like a possibility too. If the cell wants to make a copy of its DNA, why doesn't it just hang on to the DNA that it has and make a new copy of this DNA? So this ends up two copies. Well, now what we're saying is this doesn't happen. This one doesn't happen, but this one does. DNA replication happens this way. But these two scientists, they wanted to prove that this is the way it actually happens. And they set about making up an elaborate experiment. Well, it seems elaborate. Maybe it's not too elaborate. They came up with an experiment. Now, students worry about this questions to do with this experiment, and you frequently get such questions. And if you look there, you'll see I have in the booklet there. I have three such questions. Question 4, question 5, and question 6. Question 6 being the most recent example of such a question. Each of these questions essentially is about this experiment that these guys, Messes and Stahl, did to prove that DNA replication was semi-conservative. Now, as I say, the experiment is actually very complicated, and there's a lot of detail and a lot of a lot of processes in it that you possibly haven't heard of before. So, best way to approach these questions is to look at all of them and see what sorts of things the examiners ask. Before I tell you about the process, examiners tend to start these questions off. I'm looking at question 4 here. They tend to start these questions off by asking you something fairly simple about DNA. Question 4, they're showing the structure of DNA, and they're asking what kind of bonds do you have at A and B. And at A you have hydrogen bonds, they hold the bases together. And at B you have a phosphodiester bond, PHOS, PHO, DIEST are phosphodiester bonds. And then they ask, replication of DNA is described as semi-conservative. Explain this term. For 2 marks, and you would say, in the replication of DNA, you get each new DNA molecule that's produced contains an original parent strand or a template strand from the parent DNA, plus a newly made strand made from free nucleotides. That's the definition of semi-conservative replication. So they ask that. Then, traditionally in these questions, what they do is they give a brief description of the experiment. You see it there, a brief description of the experiment that these 2 guys did. And if you look at question 5, it's very similar. Once again, they ask you about the structure of DNA. They ask you which part of the DNA molecule is shown in the structure. That's a nucleotide. And then they give a brief description here, usually about a couple of paragraphs, where they're describing what these guys, Nessels and Stahl, did. Same in question 6. You'll see at some point in that question also they give a brief description of what Nessels and Stahl did to prove the semi-conservative idea. Now, what I'm saying to you is what they actually did. You'll see there on page 8 of my notes. I kind of go through it and explain it in some detail what they did. But in an exam question, you don't need to be reading that. You know, if you're reading through that paragraph or 2 paragraphs describing what these scientists did, then you're wasting your time, because you're not going to glean anything from that paragraph of information. You should already have been aware approximately of what happens in this experiment, and you shouldn't be sitting in the exam reading the detail, because the detail is not going to help you. You actually have to just get on and answer the question, and reading the detail that they've given you there will not help you. Because you simply have to answer the questions. And students, by practicing these questions and doing them quite a lot, come to understand that. They come to understand that you don't need to stop and read that detail about what the scientists did, the detail of the experiment. You just have to be good at actually answering these questions. Now, I don't intend going through the detail. Suffice to say, on the back page of my notes there is a very good diagram that kind of describes what happens in the experiment. And you won't be asked to draw bits of that diagram, you won't be asked to explain it. As you appreciate, if you've done some of these questions usually involve you drawing or having test tubes and indicating on a test tube the position of the bacteria that are involved in the experiment. So we'll come to that in a minute or two. And so this is a particularly difficult area of the course to convince students as to what they should do. But here I'm trying to convince you this. I'm trying to tell you that what you need to do is do loads of questions. Do the questions, and then you'll see you don't actually need to know the detail of the experiment, and you won't be asked any detail of the experiment. You'll be simply asked to draw some conclusions. Now, I'm just going to quickly read through the detail, just for completeness sake. These 2 scientists, Nessels and Stahl, they wanted to prove that this is the way in which DNA replicated. And what they did was they used bacteria called E. coli, which they could grow in the agar on agar plates. And as the bacteria grow in the agar plates, when they multiply, the bacteria have to copy their DNA, have to make new copies of their DNA. And before the bacteria can copy its DNA, it has to take up bases and the other components of nucleotides from the agar in which it's grown, and it needs those to make the free nucleotides that it's going to use to make the new DNA strands. And essentially, the scientists grew the bacteria. I'll just do a wee illustration. So essentially, this is the plate. So this is a plate, and this had agar here that contained nitrogen in the form of N15, and this is referred to as heavy nitrogen. And they grew the bacteria on this plate. So say that's a bacteria, and the bacteria then multiplied and made 2 bacteria. And what they're looking at is the DNA inside the bacteria. So the bacteria would contain DNA that contained nitrogen 15 atoms. So they grew the bacteria for a while in the nitrogen 15 agar. Then what they did was they moved the bacteria to another plate that contained a different agar. And the different agar was a nitrogen 14 agar. It contained a different form of nitrogen, an isotope of nitrogen 14. So simply what they did was they moved one of these bacteria from here over to this plate. So this here is here, this is the bacteria there. And this bacteria contained its original DNA, had DNA with nitrogen 15. And essentially what they want to do is they want to see what is the first generation of bacteria that's produced here. So this bacteria divided and made 2 bacteria. So there's the, I'm just making them very big there. Now, what they discovered was that these 2 bacteria, each of them contained a strand from the parent, which is nitrogen 15, but the other strand contained nitrogen 14. In other words, when this bacteria that contained 2 DNA strands that contained nitrogen 15 atoms in its bases was allowed to replicate or multiply in the nitrogen 14 agar, it was discovered that the first two bacteria, the first generation of bacteria that produced, contained each of these first generation bacteria contained one of the strands from the parent bacteria, but the other strand was made of nitrogen 14 atoms. Therefore, it was the template strand. So the first generation of bacteria that are produced here, this is called the first generation. First generation contained DNA that was intermediate, that contained both a nitrogen 15 and a nitrogen 14 strand. And this proved that DNA replication was semi-conservative. We could see that this original bacteria that had 2 nitrogen 15 strands, when it copied its DNA, the 2 nitrogen 15 strands got apart, and the other strands were made of nitrogen 14 atoms, or contained nitrogen 14 atoms. So this is the evidence that DNA replication was semi-conservative. Now, exam questions on this tend to focus on how the scientists were able to measure whether the bacteria contained N15 or N14 strands. And they're able to do that by means of centrifuging. So if you took the original bacteria that contained the N15, N15 is heavier. DNA with nitrogen 15 atoms is heavier. And what you got was a band of a group of bacteria at the bottom here, which represented nitrogen 15, both strands being nitrogen 15, N15 and 15. But the first generation of bacteria, if we drew a test tube for them, first generation bacteria, well, they didn't contain nitrogen 15. They contained intermediates. Their DNA was somewhere there. So the combination of nitrogen 15 and nitrogen 14 strands present. In other words, it was intermediate. And this first, so just me write that, N15, N14. N15, N14. And then if you went to the next generation of bacteria, I think this is maybe as far as I go to the next generation of bacteria. This was the first generation. Go to the next generation of bacteria would contain 4 bacteria. So this one would make 2 and this one would make 2. Getting a bit scrappy here. This one would make 2 and this one would make 3. What they found was some of the bacteria were just N14 and 14. And the other 2 were N15 and 14. N15 and 14. And if you represented that in terms of what you found in the centrifuge tube for the second generation, you had a band in the center which represented the N15 and 14, but you also had a band at the top which is N14 and 14. Now, I'm sure that's a very hurried explanation of what's happening there. It's very difficult to illustrate it very well in this situation, but suffice to say, I've got very good illustrations in my notes there, and they're much more easily understood than anything I've drawn on the board. So if you want to be able to understand this properly, you probably need a set of my notes in front of you if you're having difficulty. And then the other thing is, of course, to do the questions. And if you could have the question booklets there, you'll also have a set of answers, and you'll see how the questions are done. So I'm going to, by the way, these are very likely, the questions on this DNA replication and mesosin stall experiment are extremely common and extremely likely in exam questions. So it's important that you be able to not necessarily understand the detail of the experiment, because as I say, it's very complicated detail. This is the actual experiment that did involve such things as centrifuging and isotopes and so on and so forth, and that's really beyond most biology students. But actual doing the questions is relatively straightforward once you've gone through a few of them and practiced doing them. So I'm going to leave it at that. And as I say, this DNA topic, the basic understanding of DNA is very important for A2 work, and it's and at AS1 then it's part of the biochemistry topic. Okay, thank you very much. All the best. Bye.